Artificial Intelligence Dr. www.abcpharma.net/English. User question: what are the molecular mechanisms underlying the relationship between skin barrier dysfunction, xerosis, dry skin, and chronic pruritus, itching, and how do these factors interact to exacerbate skin inflammation and pain sensations? Gemini I answer. The relationship between skin barrier dysfunction, xerosis, dry skin, and chronic pruritus is a complex interplay of molecular mechanisms, leading to a vicious cycle that exacerbates skin inflammation and pain sensations. Here's a breakdown of the key players. Skin barrier dysfunction. 1. The skin barrier, primarily composed of lipids in the stratum corneum, acts as a shield against external irritants and prevents excessive water loss. 2. Disruption of this barrier, due to genetic factors, environmental insults, or underlying skin conditions, leads to increased permeability and dehydration. Xerosis, dry skin. 1. Impaired barrier function results in excessive transepidermal water loss, TEWL, causing the skin to become dry and rough, xerosis. 2. Xerosis further compromises the barrier, allowing irritants and allergens to penetrate the skin more easily. Chronic pruritus, itching. 1. Dryness activates sensory neurons in the skin, triggering itch signals that travel to the brain. 2. Additionally, barrier disruption allows inflammatory mediators like histamine and proteases to be released, further activating itch receptors. Skin inflammation. 1. Scratching due to pruritus damages the skin barrier further, leading to inflammation. 2. The release of inflammatory mediators, cytokines, chemokines, recruits immune cells, amplifies inflammation, and sustains the itch-scratch cycle. Pain sensations. 1. Inflammation and the release of neuropeptides like substance P sensitize pain receptors in the skin, leading to pain sensations alongside itching. 2. Chronic scratching can also cause nerve damage, contributing to neuropathic pain. Key molecular mechanisms. 1. Protease-activated receptors, PARs, activated by proteases released during barrier disruption and inflammation. PARs play a crucial role in itch signaling. 2. Transient receptor potential, TRP, channels. These channels are expressed on sensory neurons and are involved in sensing temperature, itch, and pain. 3. Cytokines and chemokines. Inflammatory mediators like interleukin-31, IL-31, thymic stromal lymphopoietin, TSLP, and tumor necrosis factor alpha, TNF-alpha, play key roles in both itch and inflammation. 4. Neurogrowth factor, NGF. Elevated levels of NGF are associated with increased sensitivity to itch and pain. Exacerbation of inflammation and pain. The interplay between these factors creates a vicious cycle. Dry skin leads to itching, scratching leads to barrier damage and inflammation further worsens dryness and itching. This chronic cycle perpetuates skin damage, inflammation, and both itch and pain sensations. Therapeutic implications. Understanding these molecular mechanisms is crucial for developing effective treatments. Therapies that aim to 1. Restore the skin barrier, using emollients and moisturizers to replenish lipids and prevent water loss. 2. Reduce inflammation. Topical corticosteroids or calcineurin inhibitors to suppress the immune response. 3. Target itch and pain pathways. Antihistamines, TRP channel blockers, or NGF inhibitors to alleviate symptoms. By targeting the underlying mechanisms, we can break the itch-scratch cycle, reduce inflammation, and improve the quality of life for individuals suffering from dry skin and chronic pruritus.